The aim of this movie is to introduce our present review published in Trends in Genetics, Advances using Single Particle Trajectory to Reconstruct Chromatin Organization and Dynamics, written by Ophir Shukron, Andrew Sieber, Asaf Amitai, and myself. So, electron microscopy and super resolution has revealed a static picture of chromatin. So here we can see the electron microscopy, where E-chromatin and heterochromatin are shown respectively in white and black, while here we see an example of a super resolution microscopy showing the distribution of nucleosome at a nanometer resolution. However, these pictures do not reveal the dynamics of chromatin. So, in the last couple of years, single particle trajectory of tagged locus at specific location on chromatin has been used to generate trajectories here shown in red and blue. And we have another example here where in blue is shown in yeast the single pole body, and in these two different colors, green and red, two locus positions located nearby. And one of the major challenges in the field today is to obtain an interpretation of this trajectory in terms of how chromatin is moving, what's its dynamics, and what can be extracted from this single particle trajectory in terms of the physical forces and the biophysical parameters. So if we call x of t the trajectories, it is possible to estimate the mean square displacement, that is xt plus capital T minus xt. And when capital T is small, for all t, if the process is sufficiently stationary, there's some stationarity here, it behaves like t to the power alpha. And it has been revealed that in the context of stochastic processes, that for alpha equals 1, this is usually associated to the classical Brownian motion, while alpha larger than 1 means that the process x of t contains a deterministic force, while alpha less than 1 is always associated with a motion confined to confined motion. However, we would like to revisit this concept and show that we can use more biophysical parameter to describe these trajectories. So this review is organized as follows. First part, how to extract parameters from these stochastic trajectories. Second, what is the influence of the sampling time delta t at which the trajectory are acquired. Finally, how we can use polymer models to interpret this data. Part 4. What about looking at two loci at the same time? What kind of correlation information can be obtained that cannot be found from a single loci alone? Finally, how can we reconstruct the local chromatin from associated to double-strand DNA break? And the last part is about available software that we are not going to discuss here, but this is the last part of the review. So, for parameter analysis, once a locus has been tagged, trajectory are generated, and it is possible to extract from this trajectory the distribution of the diffusion coefficient, the local diffusion coefficient, which is actually the distribution of displacement. A second interesting parameter is the length of constraint, LC, which reveal basically the region where the trajectory is confined, although it doesn't really capture the anisotropy, the possible anisotropy of this distribution. From the mean square displacement, it is possible to extract the anomalous exponent alpha by choosing a correct range of time, not too short and not too long, where the behavior is usually described or predict as a diffusion. The anomalous diffusion only occur in a certain regime of time. Finally, it is possible to extract when a trajectory is confined, whether or not it is due to the presence of a force associated to a potential well here. And the difficulty or the challenge is to extract what is the spring constant Kc. So the empirical estimators, it is possible to write for these 4 parameters some estimators that are summarized here. So for the effective diffusion coefficient, this following expression is used. It is associated to the second moment computed directly on point of the trajectories. The spring constant Kc is related to the trajectory according to this formula, where we calculate the difference between what happened at time t and t plus delta t divided by this quantity. The length of constraint is usually calculated after having estimated the center of mass of the trajectory and looking at the difference between any of 2 successive points compared to this center of mass. And it is also possible to calculate the autocorrelation function. So this is an example of a trajectory, and from this experimental trajectory, once the MSD is plotted here, it is possible to fit this a to t to the power alpha and to extract alpha. Again, the difficulty is to choose the right place, the right range of time where this is going to be estimated. And this is compared here to the case of simulations, where simulation allows us to estimate this mean square displacement over a much longer range of time. So what is usually a usual practice is to calculate, to fit using the log-log plot in order to differentiate the different time regime and to fit in the right time scale. So all of this is described in the review. So for the spring constant, one possibility is not to calculate simply this ratio, which might be problematic when the points are very close to the center. Instead, it is better to look at the distribution of xt plus delta t minus xt here versus the denominator quantity, and just to use the regression method to fit a line that will precisely give us an estimation of the spring constant as shown here. Polymer models. So there are many polymer models available, but I think one of the most important one is a class associated to the Ross polymer. And so the Ross polymer consists of beads that are connected by spring, and it is possible to add different forces such as the Lennard-Jones forces, binding forces on this. A second type of polymer that has been used recently is the beta polymer model, where forces are added between the different beads here so that the anomalous exponent is prescribed, and so the method allows to describe how to put these different long-range forces. The bead and spring model consists of a model of Ross type, where in addition binding molecules can move and can bind to a specific site, bringing together 2 parts of the polymer. Finally, the random crosslink polymer uses the same idea, but instead of having moving molecules, crosslinkers are added between 2 points of the polymer, so bringing together again 2 parts that are not necessarily close by. What's the influence of the sampling time delta t? So once a spot is chosen and the sample time delta t can change from 80 milliseconds to 300 milliseconds, we have represented here the sphere of influence. That is, during 80 milliseconds, only a certain fraction of the polymer model might influence the given locus, while for 300 milliseconds, this radius has increased. And this effect can be seen when looking at the distribution of the different parameters, such as alpha, D, L, C, and Kc. And here we summarized, in the case of yeast, for different delta T of 80 and 300 milliseconds, what are the changes that actually occur by changing this time step delta T. So we discussed specifically this part in the review, and how and this allows to understand that by changing delta T, this parameter can vary. So another possibility discussed in the review is to have an interpretation of the anomalous exponent alpha. And so this is an example through the beta polymer model where alpha varies from 0.5, which is classical Ross, and decreases to 0.4 or 0.3. And these changes in alpha is associated to a condensation of the polymer from characterized by a decrease of the radius of gyration from 2 to 1 to 1.21, showing a decrease in the radius of gyration, and thus the polymer is condensed. So so we conclude that alpha provides an information about the level of condensation of the chromatin. Another important use of this parameter is to describe confinement. So we can imagine that here a polymer model is confined between two chromosome territories. So chromosome territories can lead to confinement, but also adding cross-linker can also lead to the same effect of confinement. So how can we differentiate between the two? So although this is today still an open question, this is also discussed in the review. So in the next slide, we discuss now what the information can be recovered for monitoring two loci at the same time. So this is an example of chromosome 14 where two specific loci are tagged, and trajectory of these two loci can be generated. So although it is possible to apply the analysis of the 4 parameters to each loci separately, it is also interesting to look at these two loci together and specifically look at the distance between them and how it varies with time. So for this, the distance between the two loci can be calculated at time t. A threshold epsilon can be chosen. So here this is an example of 300 nanometers. And then the following statistical quantity can be collected. What is the time where the distance is above this 0.3 micrometer or below? So this is here an example of these sequences that can be collected into a sequence of time. And here we will recall the case where the distance, the genomic distance between those two loci can vary across experiments from 25 to 100 kilobase pairs. So again, the two loci here are shown. They are associated to trajectories. And so we looked at the time that they are staying inside these balls and the time that they are staying in between in this yellow region. And once we collected this time, they can be plotted. And what is interesting is that they are well approximated by a single exponential for the two cases, for the time the association time, which is the time the two loci takes to come into this ball, and the dissociation time, the times once they are inside the ball to escape these confined regions. And so in the review, we recall that this analysis has been done for different distances, genomic distances between these loci, such as 100, 70, and so on. And so when this association and dissociation time are then plotted versus this encounter distance epsilon, what has been found in the literature is that for the case of an untreated versus Eocene, which induced double-strand DNA break, the association time was almost not changing, except in a region of 200, 250 nanometers, where it takes more time for Eocene to associate it, probably because because probably there was a reorganization of the chromatin, and in the case of double-strand DNA break, the chromatin has been decondensed. And this result is also confirmed here in the dissociation time, the time it takes for these two loci to move apart. So it takes less time for Eocene, again, probably because locally the chromatin is less crowded. So again, this analysis shows how two spots can be used to reveal more information that were not contained in this single spot analysis. So to finish this review, we discuss modeling chromatin organization and how polymer model has been used for this. So by adding connectors here on the Ross polymer, chromatin or the polymer model becomes more condensed. Then to simulate double-strand DNA break, the link between monomer 50 and 51 is removed, as well as the green connectors. And so then, how is it possible to connect these two data, such as if we know that the length of constraint is 0.13 before and move to 0.23 micrometer after Eocene? How does this translate into a number of connectors? And particularly, how many connectors needs to be removed to go from these two lengths? And we've shown that you need quite a bit of connectors, although not a lot needs to be removed, at least locally, to obtain this calibration to data. So this is an example where the polymer models allow to estimate the distance, the mean distance between these two monomers in the case of unbroken and after a double-strand DNA break. The radius of duration can be calculated for this monomer as a function of this added connector, as well as the encounter time between the two, showing that it can vary here from 1 to about 3 seconds. So to conclude, we've seen that from single particle trajectories in different conditions, such as in normal condition or after double-strand DNA break, that the change in the single particle trajectory reveal information about chromatin reorganization that can be used to constrain polymer models. And these polymer models are used to represent chromatin and to obtain information about the statistics. So the protocol is the following. After the acquisition of trajectories, now software are available to extract parameters such as the length of constraint, the diffusion, effective diffusion coefficient, the mean spring force Kc, or the anomalous exponents alpha. They are used to constrain polymer models, and from these polymer models, it is possible to perform simulations And these simulations allow us to obtain specific information about statistics or other parameters that are not contained in the single particle trajectories. And to finish, I would like to show this movie where we have looked into the simulations of two TADs, where here you see in red the in green where we have the connectors. So at a certain point UV damages are induced, leading to an extension. So this is still in the normal period. So you see how they move using these stochastic simulations. Yeah, and suddenly you see the expansion, and the expansion is the for this we have removed locally some connectors, and then we let the system evolve by adding connectors, and you see these two blobs which can be interpreted as two TADs, how they evolve and how they start to shrink again. So I thank you for watching this video which summarizes our review.